Okay, a molecular biologist is investigating the mechanism of cellular aging and discovers this ribonuclear complex with reverse transcriptase activity. And it's going to extend the 3' ends of the chromosome by adding these repeating nucleotide sequences. She isolates a cell from a 75-year-old individual and identifies these cells exhibit significantly higher levels of this enzyme than most. The researcher hypothesizes that the cells have extended proliferative capacity to do what? All right, so it's the 3' end. So these are going to be the telomerase. Yeah, we're going to be elongating the telomeres so that these epidermal basal cells can grow and last many cycles of transcription. Okay, so here's an example of telomerase. It's a separate enzyme that works outside the normal DNA replication and extends the chromosomes. So normally DNA replication can't fully copy the ends of chromosomes, and so it gets shorter every time. An example is like a pencil sharpener. It gets shorter every time, and so telomerase is like adding wood or graphite so that the pencil never runs out. It's used for stem cells and germ cells, and most body cells like the heart, brain, and pancreas are low or have no telomerase, so they'll eventually stop dividing. Cancer cells hijack telomerases so that they can just divide forever. So telomerase is separate from regular DNA copying, but it keeps your cells young and dividing. So this is telomerase, and they'll add the TTAGGG repeats, and it's composed of telomerase reverse transcriptase, TERT, which this has a DNA and RNA template provided by the RNA component TERK. So stem cells are going to have a lot of these telomerases because they have to continually renew, and it prevents premature aging. So epidermal basal cells is the correct answer because it's part of the regenerative layer. So it's just about knowing which of these cells divide and which don't. Neurons are postmitotic. Erythrocytes do not have nuclei or DNA. Pancreatic beta cells are already distinguished enough, and they have limited regenerative capacity, so they don't exhibit significant telomerase activity. Cardiomyocytes are non-proliferative in adults. Following injury, they do not regenerate efficiently, and instead they rely on fibroblast-mediated scar formation. So hopefully that was helpful, just a little illustration of telomerase and how it might be tested on step 1.